Alright friends, welcome back to another video session from this channel. So in this video session, we are going to learn about one of the important life process which is responsible for sustaining of life on the earth or for the existence of life on the earth. And it is nothing but what? The translation mechanism or the translation process, right? So let me write it on the board. What it is? Translation mechanism. Translation mechanism or the translation process. So here at first we should have a basic idea regarding the translation mechanism or the translation process. So what exactly the translation mechanism or the translation process is? So here before studying the translation mechanism or the translation process in detail, at first we should have a basic idea regarding the central dogma of molecular biology. So according to the central dogma of molecular biology, when a particular gene, when it participates in the gene expression, it synthesizes a single stranded RNA, right? When a particular gene, when it participates in the gene expression, it synthesizes a single stranded mRNA. And once when the specific mRNA, when it participates in the translation mechanism or the translation process, it synthesizes a polypeptide chain. So let me write it here. A particular gene, when it participates in the transcription mechanism, what it is? Transcription mechanism, it synthesizes a single stranded RNA, right? And once when the specific mRNA, when it participates in the translation mechanism, what it is? Translation mechanism or the translation process, it yields or it synthesizes a polypeptide chain or a protein. So the specific mechanism or the specific process through which the single stranded RNA or the single stranded mRNA, it is capable to yield or it is capable to synthesize the polypeptide chain or the protein. It is nothing but what? Translation. So by taking the advantage of this specific mechanism or the specific process, here the synthesis of the proteins takes place in case of prokaryotic organism and as well as in case of the eukaryotic organism. So here the entire mechanism or the entire process of the translation, it can be explained under three different phases. So what are the three different phases here? The three different phases include initiation phase, elongation phase and the termination phase. So the entire mechanism of the translation mechanism, it can be explained under three different phases, and these include initiation phase, elongation phase and the termination phase. So here before studying the entire mechanism or the entire process of the translation in detail, at first we should have a basic idea regarding the various raw materials which are required for organizing the translation mechanism or the translation process. So what are the various raw materials which are responsible for organizing the translation mechanism or the translation process? So the various raw materials which play a prominent role are the various raw materials which govern the translation mechanism or the translation process include mRNA, tRNA, ribosomes and amino acid tRNA synthetase. So for your better understanding, let me present it on the board. What are those? Raw materials, right? Raw materials which are required for organizing the translation mechanism or the translation process. The very first one is mRNA. The next one is tRNA. The next component is, or the next raw material is nothing but ribosomes. And the last one is amino acid tRNA synthetase. tRNA synthetase. So at first we are going to study about the structure of the mRNA. Then after we are going to study about the structure of the tRNA, followed by ribosomes and as well as amino acid tRNA synthesis in detail. So if we consider the specific mRNA, so what is mRNA here? As I have already told you at the beginning of the class, when a particular gene, when it participates in the gene expression, it synthesizes a single stranded mRNA, right? This particular gene, when it participates in the gene expression, it synthesizes a single stranded mRNA. Now the specific single stranded mRNA which is synthesized during the transcription mechanism or the transcription process, it is quite complementary to the template strand or the antisense strand of the DNA. So as we all know, DNA is double stranded, right? So out of the two strands of the DNA, so one of the particular strand, it acts as a template DNA for the synthesis of the mRNA during the transcription mechanism. Now the specific mRNA which is synthesized during the transcription mechanism or the transcription process, it is quite complementary to the antisense strand or the template strand. So for your better understanding, let me draw the diagram of mRNA, right? So that easily you can understand. This is the structure of the mRNA, right? So let us assume it is the 5' end. Let me use some other color here. So here, this is the structure of the mRNA, right? Structure of mRNA. So the specific mRNA which is synthesized during the gene expression, it is quite complementary to the antisense strand or the template strand of the DNA. So here we need to understand one key point here. The specific mRNA which is synthesized during the gene expression or during the transcription mechanism by taking the advantage of an enzyme known as what? RNA polymerase 2. RNA polymerase 2, it is the enzyme which is responsible for synthesizing the mRNA, right? So during the transcription mechanism or the transcription process, RNA polymerase 2, it is the enzyme which is responsible for the synthesis of the single-stranded mRNA. Now the specific single-stranded mRNA which is synthesized during the transcription mechanism or the transcription process, it consists of a series of triplet nucleotide sequence or the codons, right? The entire mRNA which is synthesized during the transcription mechanism by taking the advantage of an enzyme known as what? RNA polymerase 2. It contains a series of triplet nucleotide sequences which are present very adjacent to each other. So this specific triplet nucleotide sequence which is present or which is encoded in the mRNA, it is nothing but what? Codon, right? So let me write it here. What it is? Codon. So let us assume here there is an availability of one codon, AUG, GUG, GUC, CUC and AUC, right? So let us assume there is an availability of some codons here. So here the triplet nucleotide sequence, it is nothing but what? Codon. Triplet nucleotide sequence, it is nothing but what? Codon. So here the specific codon which is a part of the mRNA, it is very specific to one particular amino acid. So as we all know in nature, there is an availability of 20 amino acids, right? So each and every amino acid which do exist in nature, it is very specific to single particular codon which is present in case of the mRNA. So here we need to understand one more key point. What it is? As I have already told you, the specific mRNA which is synthesized during the transcription mechanism by taking the advantage of RNA polymerase 2, it contains a series of triplet nucleotide sequence which is nothing but what? Codons, right? So here the ribosomal subunit which is responsible for the synthesis of the protein, the ribosomal subunit which is responsible for the synthesis of the protein, it is capable to study the biological information which is present in case of the mRNA only at this particular region, right? The ribosomal subunit, it is capable to study the biological information which is present in case of the mRNA only within this particular region. But whereas the ribosomal subunit, it is unable to study the biological information which is present in case of the mRNA at the extreme 5' and the extreme 3', right? Ribosomal subunit which gets recruited to the mRNA, it is capable to study the biological information which is present only within this region, but it is unable to study the biological information which is present at the extreme 5' and as well as the extreme 3'. So such specialized component of the mRNA which is unable to study or which is unable to encode by the ribosomal subunit, it is known as what? 5' UTR. What it is? 5' UTR and 3' UTR. So here, what is the meaning of UTR here? That means the ribosomal subunit, it is capable to encode the biological information only within this region, but it is unable to study the extreme 5' end. So the reason why it is abbreviated as what? 5' untranslated region. In the same way, at the extreme 3' end, the ribosomal subunit, it is unable to encode the biological information. So the reason why it is abbreviated as 3' UTR. The untranslated region, it is present both at the 5' end and as well as at the 3' end. So here we need to understand one more other important key point which is present in case of the mRNA. In case of the mRNA, either in case of the prokaryotes or in case of the eukaryotes, the specific mRNA, it contains the specific codons which are present here. The specific mRNA, it contains a series of codons. The specific mRNA not only contains a series of codons, in addition to the series of codons, the specific mRNA, it also contains some stop codons or termination codons, right? The specific mRNA, it not only contains a series of codons which are encoded here, in addition to the series of sense codons, the specific mRNA, it also possesses or it also contains stop codons or the termination codons. The ribosomal subunit, it is capable to study only the sense codons which are present in case of the mRNA, and it is unable to study the stop codons or the termination codons which are present in case of the mRNA. And here we need to understand one of the important difference in between the prokaryotic mRNA and in case of the eukaryotic mRNA. In case of the prokaryotic mRNA, the specific prokaryotic mRNA, it contains only the sense codons which are present very adjacent to each other. But whereas in case of the eukaryotic mRNA, two successive sense codons, they get separated by the means of the specific non-coding sequences which are nothing but what? Introns. That means in case of the eukaryotic mRNA, in between two successive sense codons, there is an availability of what? Non-coding sequences, which are nothing but what? Introns. So the non-coding sequences are the introns which are present in case of the eukaryotic mRNA. They get successfully spliced out from the particular eukaryotic mRNA by the means of a specific mechanism or by the means of a specific process, and it is nothing but what? RNA splicing. What it is? RNA splicing. RNA splicing, it is the mechanism which is responsible for splicing out the non-coding sequences which are present in between two successive sense codons. So here the mRNA which is available in case of the prokaryotic organisms, the specific mRNA which is available in case of the prokaryotic organisms, it is considered or it is defined as what? Polycistronic mRNA. But whereas the specific mRNA which is available in case of the eukaryotic organism, it is considered as what? Monocistronic mRNA, right? So let me write it here. In case of prokaryotes, the specific mRNA which is available is polycistronic mRNA, right? Polycistronic mRNA. And whereas in case of eukaryotic organisms, the specific mRNA which is available is monocistronic mRNA. So here we need to understand the variation which is present in case of the polycistronic mRNA and as well as the monocistronic mRNA. So why the particular mRNA, why it is considered as polycistronic mRNA, and why the particular mRNA in case of eukaryotic organisms, it is considered as monocistronic mRNA? The answer to this question is very simple. In case of prokaryotic organisms, the genome complexity is too less, right? In case of prokaryotic organisms, as we all know, the genome complexity is too less. So all the genes which are present in case of the prokaryotic genome, once when they undergo expression at a particular time, it leads to the synthesis of a specific mRNA, and it is nothing but what? Polycistronic mRNA, right? So let me present it here. So let us assume it as the prokaryotic genome, right? Prokaryotic genome. So as we all know, what is the genome here? What is the meaning of a genome? A genome is nothing but what? It is an association of, or it is a set of n number of genes which are present in case of an organism. The n number of genes which are present in case of an organism, they get associated in the means of a specific genome. So once when n number of genes which are present in case of the prokaryotic genome, so once when they undergo expression at a particular time, it lead to the synthesis of a specific mRNA, and that specific mRNA, it is nothing but what? Polycystonic mRNA. What it is? Polycystonic mRNA in case of prokaryotes. Polycystonic mRNA. But whereas in case of the eukaryotes, the scenario is quite opposite. In case of eukaryotes, let us assume it as the eukaryotic genome, right? Eukaryotic genome. So once when the eukaryotic genome which consists of n number of genes, in case of eukaryotic organisms, all the genes which are present in case of the genome, they won't undergo expression at a particular time. Are you able to understand? In case of eukaryotic organisms, all the genes which are encoded in case of the particular genome, they won't undergo expression at a particular single time. Each particular gene which is present in case of the eukaryotic genome, they undergo expression individually. So the reason why the specific mRNA which is available in case of the eukaryotes, it is considered as what? Monocystonic mRNA. So each particular gene when it undergo expression during the transcription mechanism, it leads to the synthesis of a single stranded mRNA. It leads to the synthesis of a specific mRNA, and that specific mRNA, it is nothing but what? Monocystonic mRNA. So right, in case of prokaryotic organisms, the specific mRNA which is available is polycystonic mRNA, and whereas in case of eukaryotic organisms, the specific mRNA which is available or which do exist is monocystonic mRNA. In case of prokaryotes, it is polycystonic mRNA, but whereas in case of eukaryotic organisms, it is monocystonic mRNA. So this is about the mRNA, right? So the mRNA structure and the various nucleotides which are encoded in case of the specific mRNA, and the variation which is present in case of the prokaryotic mRNA and as well as the eukaryotic mRNA. So the next specialized raw material which is responsible for organizing the translation mechanism or the translation process, it is nothing but what? tRNA, right? So let me present it here. What it is? tRNA. tRNA is the next specialized raw material which is responsible for organizing the translation mechanism or the translation process. So here now my question is, before studying about the tRNA structure in detail, at first let us have a basic idea regarding the structure of the tRNA. Why the particular tRNA? Why it is considered as tRNA? Why the name tRNA is given to the particular RNA? The name tRNA is given to the particular RNA because the structure of the RNA, it resembles the shape of an alphabet known as what? T. So due to the resemblance of the particular RNA structure in the means of an alphabet known as T, the specific RNA, it is known as what? tRNA. So here tRNA, or it is otherwise known as transfer RNA. What it is? Transfer RNA. So what is the specific enzyme? Which is responsible for the synthesis of the tRNA? In what way the specific mRNA it is synthesized? By taking the advantage of an enzyme known as RNA polymerase 2. In the same way, there is a requirement of a specific enzyme, and it is nothing but what? RNA polymerase 3. RNA polymerase 3, it is the enzyme which is responsible for the synthesis of the tRNA. What it is? RNA polymerase 3. So let me write it here. What is the enzyme which is responsible for the synthesis of the tRNA? RNA polymerase 3. Now here my question is, what is the role of tRNA? What is the role of tRNA in case of the translation mechanism or the translation process? So here the specific tRNA, it acts as an adapter RNA molecule, right? The specific tRNA, it acts as an adapter RNA molecule. So for the recognition of the specific codons, which are present in case of the mRNA, the translation mechanism or the translation process, it takes the advantage of a specific adapter RNA molecule, and that specific adapter RNA molecule, it is nothing but what? tRNA. That means, as we all know, in nature there is an availability of 20 amino acids, right? In nature there is an availability of 20 amino acids. So each and every amino acid which do exist in nature, it is very specific to one particular codon which is present in case of the mRNA. But the specific amino acid which do exist in nature itself, it is unable to recognize the codon which is present in case of the mRNA. The amino acid which is available in case of the nature, it is very specific to one particular codon. But the specific amino acid itself, it is unable to recognize the codon which is present in case of the mRNA. So under such condition, the specific translation mechanism or the translation process, it takes the advantage of a specific RNA molecule, and that specific RNA molecule, it is nothing but what? Adapter RNA, or it is otherwise known as what? tRNA. So by taking the advantage of the tRNA, the specific amino acid which is very specific to one particular codon, it is capable to engage, or it is capable to recognize, right? So for your better understanding, let me present it here. So let us assume it as the mRNA, right? mRNA. So here there is an availability of some codons like AUG and GUG, right? So let us assume like that. And here there is an availability of an amino acid. So all the amino acids which do exist in nature, they are very specific to one particular codon which is present or which is encoded in case of the mRNA. But the specific amino acid itself, it is unable to encode, or it is unable to recognize the codon which is present in case of the mRNA. So under such condition, the amino acid, it takes the advantage of an adapter RNA molecule, and that specific adapter RNA molecule, it is nothing but what? tRNA. So by taking the advantage of a specific adapter RNA molecule known as tRNA, the specific amino acid, it is capable to recognize the codon which is present in case of the mRNA, right? So here we need to understand the structure of the tRNA. So what are the various components which are present in case of the tRNA? So if you are capable of understanding the various components which are a part of the tRNA, then easily you can understand the structure of the translation mechanism or the translation process in detail, right? So let me present the structure of the tRNA here. So just I will draw the rough diagram so that easily you can understand here. So let us assume it is the tRNA, right? So just I am showing you the rough diagram here. Just around like this. Something around like this, right? And here there is an availability of acceptor stem, ACC, and it is the 5' end. And here there is an availability of anticodon loop. What it is? Anticodon loop, and it is nothing but the D loop here. D loop. Let me erase it. D loop, and it is nothing but the T-C loop, right? And it is nothing but the variable loop. So here all the nucleotides are encoded here within the tRNA. So all these are the various nucleotides which are encoded in the particular tRNA, right? So now here my question is acceptor stem, right? So here now my question is, what is the role of individual components which are present in case of the particular tRNA, right? So here why the particular stem which is present in case of the tRNA, it is considered as acceptor stem. So the particular component which is present in case of the tRNA, it is considered as an acceptor stem because the specific amino acid, as I have already told you earlier, each and every amino acid which do exist in nature, it is very specific to one particular codon which is present in case of the mRNA, right? But the specific amino acid itself, it is unable to recognize the codon which is present in case of the mRNA. So that the specific amino acid, it takes the advantage of an adapter RNA, and that specific adapter RNA, it is nothing but what? tRNA. So here the amino acid which is very specific to one particular codon, it is capable to encode or it is capable to engage with a particular tRNA. So where exactly the particular amino acid it gets engaged with the particular tRNA? The particular amino acid, it gets engaged with a particular tRNA by taking the advantage of a specific site or by taking the advantage of a specific stem. And that specific stem, it is nothing but what? Acceptor stem. So by taking the advantage of a specific stem known as acceptor stem, which is present in case of the tRNA, each and every amino acid which do exist in nature, it is capable to recruit or it is capable to engage with the tRNA, right? So it is nothing but what? Acceptor stem. So what it is? Acceptor stem. So in addition to the acceptor stem, there is an availability of variable loop. What it is? Variable loop. So why the particular loop? Why it is known as variable loop? Because the size of the variable loop, it won't remain similar nearly in case of the all organisms or nearly in case of the all the species. The size of the particular loop, it gets varied from one particular organism or from one particular species to the another species. So the reason why the particular loop, it is considered or it is defined as what? Variable loop. So in addition to the variable loop, there is an availability of one more other specialized loop, and it is nothing but what? Anti-codon loop, right? So it is nothing but what? Anti-codon loop. So why the particular loop? Why it is considered as anti-codon loop? Because as I have already told you, each and every amino acid, it is very specific to one particular codon of the mRNA. So here to engage with one particular codon which is present in case of the mRNA, right? So each and every amino acid, it gets recruited to the particular tRNA at a specialized site known as acceptor stem, right? Or the acceptor site. So after the successful recruitment of one particular amino acid to the acceptor stem or acceptor site, now the specific tRNA, it is ready to participate in the translation mechanism, and the specific tRNA, it is capable to study the codon which is present in case of the mRNA. So here, in order to recognize the codon which is present in case of the mRNA, the specific tRNA, it takes the advantage of a specialized loop, and it is nothing but what? Anti-codon loop. So the specific anti-codon loop, which is a part of the tRNA, it is quite complementary to the codon of the mRNA, right? So let us assume here there is an availability of codon known as what? AUG. So in return, in case of the tRNA, it should be 3' to 5', right? So here in case of the mRNA, let us assume there is an availability of one particular codon known as AUG. So here the complementarity of G is nothing but what? C. The complementarity of U is A, and the complementarity of A is U. So here it is nothing but what? Codon. It is nothing but what? Codon of the mRNA. So the codon of the mRNA, it is quite complementary to the anticodon of the tRNA, right? So in order to recognize the codon which is present in case of the mRNA, the specific tRNA, it takes the advantage of a specialized loop, and that specialized loop, it is nothing but what? Anticodon loop. So in addition to the various components of the tRNA, the specific tRNA, it also possesses or it also contains some unusual bases, right? The specific tRNA, in addition to the usual bases like AUGC, what are those? AUGC. AUGC. As we all know, in case of the RNA, there is no availability of thymine, right? So instead of thymine, there is an availability of uracil here. So in case of the RNAs, like it may be either rRNA or mRNA, or it may be either tRNA. In case of all the RNAs, there is an availability of usual bases like AUGC. So in addition, in case of the tRNA, in addition to the usual bases like AUGC, there is an availability of unusual bases. What are those unusual bases? In case of the tRNA, in addition to the usual bases like AUGC, there is an availability of unusual bases here. So what are the various unusual bases which are a part of the tRNA structure? The various unusual bases include, let me write it here. What are those? Uridine. What it is? Uridine. Pseudouridine. The next one is dihydrouridine. Ribothymidylate, and cytidylate, hypoxanthine, and xanthine. So right. So in addition to the normal usual bases like AUGC, in case of the tRNA, there is an availability of some unusual bases, and these unusual bases include uridine, pseudouridine, dihydrouridine, ribothymidylate, cytidylate, and hypoxanthine and xanthine, right? So all these are the unusual bases which are present in case of the tRNA. And all these unusual bases, they won't get recruited into the tRNA structure during the transcription mechanism, right? The normal usual bases like AUGC, they get recruited to the tRNA structure during the transcription mechanism of the transcription process. But what about the unusual bases here? The unusual bases, they won't get recruited to the tRNA structure during the transcription mechanism. After the successful synthesis of the tRNA during the transcription mechanism by taking the advantage of an enzyme known as RNA polymerase 3, now the specific tRNA, it undergoes a specialized event, and that specialized event, it is nothing but what? tRNA processing. So let me write it here. What it is? tRNA processing. So during the tRNA processing event, the usual bases which are present in case of the tRNA, during the tRNA processing event, the usual bases which are present in case of the tRNA, they undergo certain modifications, so that the usual bases, some of the usual bases, they are modified or they are replaced by the unusual bases in case of the tRNA, right? So after the successful synthesis of the tRNA, during the transcription mechanism or the transcription process by taking the advantage of an enzyme known as RNA polymerase 3, now the specific tRNA, it undergoes a specialized event, and it is nothing but what? tRNA processing. So during the tRNA processing event, some of the usual bases which are present in case of the tRNA, they are modified by unusual bases. So here the unusual bases, they get recruited to the tRNA structure during the tRNA processing event. Even the acceptor stem, which is present at the 3' end of the tRNA, it is not a part of the tRNA gene. The specific trinucleotide sequence, it gets successfully recruited to the tRNA structure during the tRNA processing event. So for the recruitment of the trinucleotide sequence at the 3' end of the tRNA, there is a requirement of an enzyme known as what? Terminal nucleotidyl transferase. So by taking the advantage of an enzyme known as terminal nucleotidyl transferase, the trinucleotide sequence, it gets successfully recruited to the tRNA structure. So this is about the tRNA structure, and the specific tRNA which participates in case of the translation mechanism or the translation process. Generally they are of two different types, right? The specific tRNAs which participate in case of the translation mechanism or the translation process, Generally, they are of two different types. So what are the two different types of tRNAs here? The two different types of tRNAs include, one is initiated tRNA and the other one is amino acid tRNA, right? So let me present it here. So what are the two different types of tRNAs which are present in case of the translation mechanism or the translation process? One is initiated tRNA. One is initiated tRNA. And the other one is amino acid tRNA. Amino acid tRNA. So one is initiated tRNA and as well as amino acid tRNA. Now here my question is, how the translation mechanism or how the translation process it is capable to differentiate or it is capable to distinguish that it is the initiated tRNA and it is the amino acid tRNA. So in order to distinguish the initiated tRNA from the amino acid tRNA, the translation mechanism or the translation process, it takes the advantage of some unique features, right? So by taking the advantage of some unique features, the specific translation mechanism or the translation process, it is capable to distinguish the initiated tRNA from the amino acid tRNA. So what are the unique features which are present in case of the initiated tRNA? There is an availability of one specific unusual base, right? There is an availability of one specific unusual base which is present at the extreme 5' and as well as the 3' end. What it is? It is nothing but what? There is an availability of one unusual base and it is nothing but what? C. And to the adjacent of C, there is an availability of one more specific nucleotide and it is nothing but what? Adenosine, right? At the 5' end, at the extreme 5' end, there is an availability of cytosine nucleotide, and the adjacent of the specific cytosine nucleotide, there is an availability of unusual base, and it is nothing but what? Adenosine. So as we all know in nature, the strongest hydrogen bonding in between two successive nitrogenous bases, it is present in between G and C, right? There is an availability of three hydrogen bonds. There is an availability of three hydrogen bonds in between G and C. So here, to the complementarity of the cytosine, there should be an availability of what? Guanosine. So instead of guanosine, there is an availability of one specific unusual base and it is nothing but what? Adenosine. Instead of the presence of guanosine here, there is an availability of adenosine. So it is one of the unique features. It is one of the unique feature which is present in case of the initiated tRNA. Only in case of the initiated tRNA, there is an availability of this unusual base pairing. So in addition to this specific unusual base pairing, even in case of the initiated tRNA, there is one more other remarkable feature. So what it is? It is nothing but presence of three consecutive GC base pairing. In addition to this specific remarkable feature, there is an availability of one more other specific unique feature which is present in case of the initiated tRNA, and it is nothing but what? Presence of three consecutive GC base pairing which is present very near to the anticodon loop. So here there is an availability of three GC base pairings, right? Three GC base pairing. So in addition to this specific remarkable feature, in case of the initiated tRNA, there is an availability of three consecutive GC base pairing very near to the anticodon loop of the tRNA. So the specific remarkable features, they are present only in case of the initiated tRNA. But whereas in case of the amino acid tRNA, there is no availability of this remarkable features, right? So by taking the advantage of this, some unique features which are present in case of the initiated tRNA, the translation mechanism or the translation process, it is capable to distinguish the initiated tRNA from the amino acid tRNA, right? So the specific initiated tRNA, it gets successfully recruited to the ribosomal subunit during the initiation phase of the translation, but whereas the amino acid tRNA, it gets successfully recruited to the ribosomal subunit during the elongation phase of the translation. So which we are going to discuss further, right? So this is about the tRNA structure. Then what about ribosomes here? What are ribosomes, or how we can define ribosomes? What are ribosomes? So actually ribosomes are nothing but what? Ribonucleoproteins, right? Ribosomes are nothing but ribonucleoproteins. Ribonucleoproteins. That means they are an association of what? RNA and as well as protein, right? They are an association of RNA and as well as protein. So the specific ribosomes, they are available both in case of the prokaryotic organisms and as well as in case of the eukaryotic organisms. Both in case of the prokaryotes and as well as in case of the eukaryotes, there is an availability of ribosomes. But the specific ribosomes which do exist in case of the prokaryotes and as well as in case of the eukaryotes, they are quite different. So what are the specific ribosomes which are present in case of the prokaryotic organisms? The specific ribosomes which do exist in case of the prokaryotic organisms, they are nothing but what? 70S ribosomes. So let me present it here. In case of prokaryotes, there is an availability of specialized ribosomes and they are nothing but what? 70S ribosomes. And the specific 70S ribosomes, they get further differentiated into 50S and 30S. So here the 50S subunit is the largest subunit and 30S subunit is the smallest subunit. In the same way, in case of the eukaryotic organisms, there is an availability of one specialized ribosome and it is nothing but what? 80S ribosome. So here, in what way the 70S ribosomes, they are further differentiated into 50S and 30S. In the same way, the 80S ribosomes, they are differentiated into 60S and as well as 40S. So here, the 60S subunit is the largest subunit and the 40S subunit is the smallest subunit, right? So here we need to understand the chemical composition of the ribosomes. So as I have already told you, the specific ribosomes, they are ribonucleoproteins. That means they are an association of RNA and protein, right? So if we consider in case of the prokaryotes, the 50S ribosomes, they are composed of what? 23S rRNA and 5S rRNA. In addition to the 23S rRNA and 5S rRNA in case of the 50S subunit of the ribosome, it is also encoded with 31 different proteins. So let me write it here. 50S subunit, it is composed of 23S rRNA and 5S rRNA. In addition to the 23S rRNA and 5S rRNA, there is an availability of 31 different proteins. And as well as the 30S subunit, the smallest subunit, which is nothing but 30S subunit, it is composed of what? 16S rRNA. The largest subunit, which is nothing but the 50S, it is composed of 23S rRNA and 5S rRNA. And in addition, it also contains 31 different proteins. And whereas the 30S subunit of the ribosome, it is composed of what? 16S rRNA. In addition to the 16S rRNA, it also possesses 21 different proteins. So this is the basic chemical composition. in case of the prokaryotic ribosomes. But whereas in case of the eukaryotic organisms, there is an availability of one more other specialized ribosome and it is nothing but what? 80S ribosomes. So 80S ribosomes, they get further differentiated into 60S and 40S, right? So here the 60S ribosome, it possesses or it contains 28S rRNA, right? The specific 60S ribosome, it possesses or it consists of 28S rRNA and 5.8S rRNA. In addition to the 28S rRNA and 5.8S rRNA, in case of the 60S subunit of the ribosome, in addition, in case of the 40S subunit of the ribosome, it also possesses 18S rRNA, right? So let me present it here. 60S ribosome, it possesses 28S rRNA and 5.8S rRNA. And whereas the 40S subunit, it is composed of 18S rRNA. So this is the basic chemical composition of the specific ribosomes which do exist in case of prokaryotes and as well as in case of the eukaryotic organisms. And here during the translation mechanism or the translation process, never and ever, both the subunits of the ribosome, they get recruited to the mRNA at a single particular time, right? Never and ever, both the subunits of the ribosome, they get recruited to the particular mRNA at a single particular time. So at the very beginning of the translation mechanism or the translation process, at first the small subunit of the ribosome, it gets recruited to the mRNA. Then after, the large subunit of the ribosome comes into the picture, both in case of prokaryotic organisms and as well as in case of the eukaryotic organisms. So here we need to understand the structural physiology of the ribosomes. So if we consider the structural physiology of the ribosome, there is an availability of 3 different sites which are present in case of the small subunit of the ribosome. So what are the 3 different specialized sites which are present in case of the small subunit of the ribosome? They are nothing but A site, P site and E site, right? So let me present it here. So let us assume it as the small subunit of the ribosome. The small subunit of the ribosome, they are composed of 3 different sites. What are those? A site, P site and E site. A site, P site and E site. So here the A site is the specialized site for the recruitment of what? Amino acid tRNA. A site of the smallest subunit of the ribosome, it is the specialized site for the recruitment of amino acid tRNA. And P site is the specialized site which is responsible for the recruitment of what? Initiated tRNA or the peptidyl tRNA. So so let us assume it as the initiated tRNA, right, with its encoding amino acid, and let us assume it as the amino acid tRNA with its encoding amino acid. So once when the specific initiated tRNA or the peptidyl tRNA which was recruited to the P site of the ribosome, after the successful participation of the particular tRNA in the translation mechanism or in case of the protein synthesis, now the specific initiated tRNA, it gets exited from the smallest subunit of the ribosome by taking the advantage of a specialized site, and it is nothing but what? E site. So E for what? Exit. After the successful participation of the respective tRNAs in case of the translation mechanism or in case of the translation process, now the specific tRNA, it gets exited from the small subunit of the ribosome by taking the advantage of a specialized site, and it is nothing but what? E site. So E stands for what? Exit. So this is about the ribosomes. Now we are going to discuss about one more other specialized raw material which is a part of the translation mechanism or which plays a prominent role in organizing the translation mechanism or the translation process, and it is nothing but what? Amino acid tRNA synthesis, right? So let me present it on the board. What it is? Amino acid tRNA synthesis. Amino acid tRNA synthesis. So let me present it on the board. What it is? Amino acid tRNA synthesis. So what is the role of amino acid tRNA synthesis here? The role of amino acid tRNA synthesis is very simple and as well as it is very complex, right? So what is the role of amino acid tRNA synthesis? As I have already told you earlier, each and every amino acid which do exist in nature, it is very specific to one particular tRNA, right? Each and every amino acid which do exist in nature, it is very specific to one particular tRNA. So here for the recruitment of one particular amino acid to its encoding tRNA, the specific translation mechanism or the translation process, it takes the advantage of an enzyme, and it is nothing but what? Amino acid tRNA synthesis. By taking the advantage of an enzyme known as amino acid tRNA synthesis, each and every amino acid which do exist in nature, it is capable to recognize or it is capable to recruit to its encoding tRNA. So let us assume it as the tRNA, right? Let us assume it as the tRNA, and let us assume it as the amino acid. So here for the recruitment of the amino acid, to its encoding tRNA, the specific translation mechanism or the translation process, it takes the advantage of an enzyme known as what? Amino acid tRNA synthetase. So by taking the advantage of an enzyme known as amino acid tRNA synthetase, each and every amino acid which is very specific to one particular codon, it is capable to engage with its encoding tRNA, right? The specific amino acid which is very specific to one particular codon, it is capable to engage or it is capable to encode to its specific tRNA by taking the advantage of an enzyme known as amino acid tRNA synthetase. So here the specific amino acid tRNA synthetase which do participate in case of the translation mechanism or the translation process, generally they are classified into two different types. So what are the two different types of amino acid tRNA synthetases here? The specific amino acid tRNA synthetases which play a prominent role for organizing the translation mechanism or the translation process, they are nothing but class 1 amino acid tRNA synthetase and class 2 amino acid tRNA synthetase. So let me write it here. What are those? Class 1 amino acid tRNA synthetase, and the other one is class 2 amino acid tRNA synthetase. Class 2 amino acid tRNA synthetase. Amino acid tRNA synthetase. So here we need to understand the difference between class 1 amino acid tRNA synthetase and class 2 amino acid tRNA synthetase. So here for understanding the activity of class 1 amino acid tRNA synthetase and class 2 amino acid tRNA synthetase, let me draw the structure of the specific purine ring, right? Structure of the purine ring, so that easily you can understand here. Amino acid tRNA synthetase. tRNA synthetase. So let us assume it has the purine ring, right? It has the purine ring. Actually, it takes some time to present it on the board. So let me draw that diagram here. Let me draw the ribose sugar. So it is nothing but the 2' hydroxyl group, and it is nothing but the 3' hydroxyl group, and it is nothing but the CH2 group here. And in addition, there is an availability of phosphate moiety, and here there is an availability of one specific amino acid. So here, as I have already told you, the specific amino acid tRNA synthetases which play a prominent role in organizing the translation mechanism are the specific amino acid tRNA synthetases which play a prominent role for recruiting one particular amino acid to its encoding tRNA. So generally they can be classified into two different types. So one is class 1 amino acid tRNA synthetase and the other one is class 2 amino acid tRNA synthetase. So what is the difference in between class 1 amino acid tRNA synthetase and class 2 amino acid tRNA synthetase? So class 1 amino acid tRNA synthetase, it recruits class 1 amino acid tRNA synthetase, it recruits one particular amino acid to the tRNA ribose sugar at the 2' OH, right? Class 1 amino acid tRNA synthetase, it recruits one particular amino acid to the tRNA ribose sugar at the 2' hydroxyl group, and whereas class 2 amino acid tRNA synthetase, it recruits one particular amino acid at the 3' hydroxyl group of the tRNA ribose sugar. So that is the basic difference in between the class 1 amino acid tRNA synthetase and class 2 amino acid tRNA synthetase. So here each and every amino acid which do exist in nature, it is capable to recruit to its encoding tRNA by taking the advantage of amino acid tRNA synthetase. That means for recruitment of 20 amino acid to its 20 tRNAs, right? For recruitment of 20 amino acids to the 20 encoding tRNAs, there is a requirement of 20 amino acid tRNA synthesis. So this is the basic concept which can be applicable to the eukaryotic organisms or in case of the major organisms. But whereas in case of the primitive organisms like bacteria, there is no availability of 20 amino acid tRNA synthesis. So under such condition, the specific amino acid tRNA synthesis which has recruited one particular amino acid to its encoding tRNA, the same amino acid tRNA synthesis, it is capable to recruit the other cognate amino acid to their encoding tRNA. So for your better understanding, let me present it on the board. One specific rule which is in association with the amino acid tRNA synthesis. So let us assume it has the tRNA structure, right? tRNA structure. And here the amino acid, it gets successfully recruited here. Amino acid, it gets successfully recruited here. It is nothing but the anticodon loop, right? So in nature, there is an availability of 20 amino acids. 20 amino acids. So here for the recruitment of 20 amino acids to their encoding 20 tRNAs, there is a requirement of 20 amino acid tRNA synthetases. 20 amino acid tRNA synthetases. So this is the basic concept, right? So this is the basic concept. For the recruitment of 20 amino acids to the 20 encoding tRNAs, there is a requirement of 20 amino acid tRNA synthesis. So this is the basic concept which can be applicable to the eukaryotic organisms or in case of the complex organisms. But whereas in case of the primitive organisms like bacteria, there is no availability of 20 amino acid tRNA synthesis. So under such condition, the specific amino acid which has recruited one particular amino acid to its encoding tRNA, the same amino acid tRNA synthesis, it is capable to recruit the other specific amino acid to its encoding tRNA. So for your better understanding, let me explain it on the board. So let us assume a specific amino acid known as glutamate, right? Glutamate, tRNA and glutamine. So when it undergoes chemical modification, it yields glutamine, tRNA, glutamine. So here we need to understand the chemical modification here. The prefix indicates what? The amino acid. Here the prefix indicates the amino acid and the suffix indicates the chemical modification. That means the specific amino acid tRNA synthesis, which has recruited the glutamate amino acid to its encoding tRNA. Once when the specific glutamate amino acid, when it was successfully recruited to its encoding tRNA by taking the advantage of one amino acid tRNA synthesis, one amino acid tRNA synthesis, amino acid tRNA synthesis, the same amino acid tRNA synthesis, it is capable to recruit the glutamine amino acid to its encoding tRNA. So how it is possible? Once when the specific glutamate amino acid, when it was successfully recruited to the tRNA by taking the advantage of the amino acid tRNA synthesis, now the specific glutamate amino acid, it possesses or it consists of amide configuration, right? The specific glutamate amino acid, it possesses or it consists of amide configuration. So once when the glutamate amino acid, when it was successfully recruited to the tRNA, now the amide configuration which is a part of the glutamate amino acid, it was successfully removed and it is modified into amine configuration, right? So once when the specific amide configuration, when it is removed and when it is replaced or when it is modified into amine configuration, now the glutamate amino acid, it is converted into glutamine. So under such condition, the specific amino acid tRNA synthesis which has recruited the glutamate amino acid to its encoding tRNA, the same amino acid tRNA synthesis, it is capable to recruit the glutamine amino acid to its encoding tRNA. So in this way, without taking the advantage of 20 amino acid tRNA synthetases in case of prokaryotic organisms or in case of the primitive organisms, the recruitment of the respective 20 amino acid to their encoding tRNA, it takes place, right? So here we need to understand one key point, what it is. In addition to the amino acid tRNA synthesis activity, the specific translation mechanism or the translation process, it also takes the advantage of the anticodon loop. Here, the recruitment of one particular amino acid to its encoding tRNA, it is not only governed by the amino acid tRNA synthetase. In addition to the amino acid tRNA synthetase activity, the anticodon loop of the tRNA, it also plays a prominent role. So for your better understanding, let me give you one common example. Right? So what it is nothing but serine amino acid. What it is? Serine amino acid. So serine is the amino acid which is capable to encode 6 codons in the mRNA, right? 6 codons in the mRNA. Serine is the amino acid which is capable to encode 6 codons. So here by taking the advantage of one specific amino acid tRNA synthetase, the specific serine amino acid, it is capable to encode 6 different tRNAs, right? Because the serine amino acid, it is capable to encode 6 codons. For recognizing the 6 codons which are present in case of the mRNA, there is a requirement of 6 different tRNAs. So here for the recruitment of 6 different tRNAs, the specific serine amino acid, it takes the advantage of only one amino acid tRNA synthetase. So by taking the advantage of one amino acid tRNA synthetase, the specific serine amino acid, it is capable to encode 6 different tRNAs. So such specific tRNAs which are capable to encode or which is capable to engage with one particular amino acid by taking the advantage of one particular amino acid tRNA synthetase, they are nothing but what? Isoacceptor tRNAs. What are those? Isoacceptor tRNAs. So here the amino acid which is recruited to the tRNA, it is only one. But the tRNAs which are present here, they are more than one, because the serine amino acid, it is capable to encode 6 codons. For the recognition of 6 codons, the specific serine amino acid, it takes the advantage of 6 different tRNAs. But here, for the recruitment of the serine amino acid to its encoding tRNA, the specific serine amino acid or the specific translation mechanism, it takes the advantage of only one amino acid tRNA synthetase. So such specific tRNAs, they are nothing but what? Isoacceptor tRNAs, right? So here the amino acid tRNA synthetase, it possesses a specialized activity, and that specialized activity is nothing but what? Proofreading mechanism or the proofreading process, right? So let me write it here. What it is? Proofreading mechanism, right? So as we all know, nearly in case of all the specific process like replication and as well as in case of the transcription, there is an availability of the proofreading mechanism. So what is the availability or what is the role of proofreading mechanism here? The specific proofreading mechanism, it plays a prominent role in the removal of the misincorporated nucleotides in case of the replication mechanism and as well as in case of the transcription mechanism. So by taking the advantage of the proofreading mechanism, or the proofreading process, the misincorporated nucleotides which were recruited during the replication mechanism or the transcription mechanism, they get successfully discarded by taking the advantage of the proofreading mechanism or the proofreading process. So such specific proofreading mechanism, even it do exist in case of the translation mechanism or the translation process. So the specific proofreading mechanism which do exist in case of the translation mechanism or the translation process, generally they are of 2 different types, right? So what are the 2 different types of proofreading mechanisms which do exist in case of the translation mechanism? They are nothing but chemical proofreading mechanism, and the other one is kinetic proofreading mechanism. So let me write it here. What are those? Chemical proofreading mechanism, and the other one is kinetic proofreading mechanism. So let me use some other color here. Kinetic proofreading mechanism. So what is chemical proofreading mechanism here? The specific chemical proofreading mechanism, it is the specialized mechanism which is responsible for the removal of the incorrect amino acid which was recruited to the tRNA. So let us assume one incorrect amino acid, it was recruited to the tRNA, right, during the translation mechanism. So under such condition, for the removal of the incorrect amino acid which was recruited to the tRNA, the specific translation mechanism or the translation process, it takes the advantage of a specific proofreading mechanism, and it is nothing but what? Chemical proofreading mechanism. So by taking the advantage of the chemical proofreading mechanism, the incorrect amino acid which was recruited to the tRNA structure, it gets successfully discarded. So it is nothing but what? Chemical proofreading mechanism. And what about kinetic proofreading mechanism? So the specific kinetic proofreading mechanism, it is a complex process. So we are going to discuss about the kinetic proofreading mechanism or the kinetic proofreading process during the elongation phase of the translation mechanism, because if I explain you about the kinetic proofreading mechanism right now, it is not so easy for you people to understand. So the reason why, when I explain you about the elongation phase of the translation mechanism, surely I will explain you what is kinetic proofreading mechanism. So all these are the various raw materials which play a prominent role for organizing the translation mechanism or the translation process. I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.